So our next speaker in the session is Dr. Rebekah Penrice Randal, and she is a research scientist at TopMD, and she'll be speaking about COVID-19 biomarker identification from multifactorial analysis.
Hello, can you hear me? Okay, so hopefully you've had a nice introduction to TopMD. So I'm going to spend some time going very briefly over some of the RNA-seq projects that I've done through Julian's lab as a postdoc, and what I'm doing now in TopMD on clinical cohorts of patients with COVID-19. So, as we all know, there is a spectrum of disease within COVID-19. So some people are asymptomatic, some people get mild disease, some people have severe disease, and these people tend to end up in hospital and it can result in fatality. We obviously have countermeasures in place, vaccines, antivirals, public health measures, which try and push us that in the opposite direction. But what I wanted to really point out here is that we can see from experience that there is huge diversity with how people experience disease. And going back onto Jim's talk about different phenotypes of disease, there's probably molecular differences going on within COVID patients. So we use RNA sequencing to look at how people are responding to COVID-19 with traditional RNA-seq analysis pipelines, such as differential gene expression analysis, but also TopMD. Most of the studies I'm going to go over now are kind of restricted to hospitalization, so people who've gone to hospital, but I will take through those anyway. So the first study I'm going to talk about is one that we started doing back in 2020 in collaboration with the University of Southampton and TopMD. And this was a point of care study where we aimed to compare the transcriptome of those with SARS-CoV-2 infection and influenza A infection at point of admission. And we had about 80 odd patients within the COVID cohort and in the influenza cohort. Now, at admission, a lot of the clinical scoring is quite similar. So we wanted to see if we could pick out any differences within the transcriptome. So one of the key findings really within this study is that I've not shown it here, but when you look at the PCAs of this data, there's a huge overlap within the transcriptomic profiles within influenza patients and COVID patients. So it's generally a interferon virus response. So the differences between these two viruses in people are quite discrete. But what we did find is that these immunoglobulin transcripts were higher in COVID patients and in those that went on to survive. So we further went on to look at what this looked like within the TopMD pathway analysis. And again, one of the key things here is that we were able to see the data from a different perspective. And one of the key things that I want to sort of mention is that we found this a set of genes enriched within the transcription factor MYC processes. And when we looked into the literature and tried to understand that a bit more, we knew that activated MYC represses IRF7, which was reduced in COVID-19 patients. So then we took the same data set and we wanted to see if we could use TopMD pathway information to predict ICU admission. So these are the top 3 pathways that came out. So here's your log2 fold changes that you might be familiar with traditional RNA-seq analysis, and these are the genes that are within the pathways. And then when we looked into the literature of what these actually meant, we found that these made a lot of sense. So actually, these are representative of EGFR signaling and TGF-beta signaling pathways. And there's been quite a lot of studies in all of the coronaviruses pretty much, that suggests that these pathways are involved in pulmonary fibrosis, which we know is associated with severe COVID-19. So I was quite happy to see that these pathways made biological sense. And within this, it was simple test and train logistic regression, and that was had a predictive power of 0.76, but we didn't have a validation data set. So that brings me onto the current project that we're currently working on, which is the Dragon Consortium, which has brought together so many different people from so many different backgrounds. So we've got people that are based in CT scanning and AI, where they take the picture of the CT scan and generate quantitative data, and from that are able to predict the severity of COVID without the need of a clinician. And I guess the whole scope of this is to empower and increase capacity of healthcare systems with AI. So our role in that really is to lead on this pathway biomarker part of things, and also bring in the different types of data that's been generated within this consortium, including this CT scan imaging. So when I was back in Liverpool, we spent a long time looking at tests, extracting the blood in the lab. We extracted a good 352 samples, and then we sequenced it on a NovaSeq Illumina platform. Sorry. So the roles here was that Liverpool/me did the like traditional sort of approaches, TopMD do pathways, and then we also had a little look at splicing from our collaborators in Southampton. So because the data, you can't really ask for your data to have the right numbers of people who are admitted to ICU, or your nice bit of information that you want to predict. Within this data set, there was quite a low number of ICU admission and mortality. So we had to be a bit creative of what we wanted to use as a predictor in this first instance, which is just sort of to take us through the data. So we had two cohorts, one from Florence, one from Liège in Belgium. And so we split the Florence data set into the training and test data set, and the Liège data into our external validation set. We chose high oxygen therapy as a proxy measurement of those who might be on a more severe trajectory, which of course has limitations, but I'll take you through at the end what we're doing to counteract that. So yeah, as I mentioned, these are what we're sort of doing on the project. Unfortunately, we didn't really find anything interesting in the splicing biomarker side of things, but we are still continuing on the traditional approaches and the TopMD analysis. So another part of our role in this consortium is pulling in this gene expression data and pathway activation data, our clinical data, and our CT scans, which at the moment are being mostly scored by a clinician, but we also work with this other biotech company called Therona, which have successfully sort of built their own model just exclusively using their data, which you can read about there. So our sort of goal there is to bring in all of this different data types to build a model which eventually might go into a bigger app or a patient-centered platform to inform clinical decisions. So our CT scan data that we're working with at the moment, we're using the CORAD scoring system, which is how likely the suspicion of having COVID from the CT images. And then we're also working with scoring systems which sort of define how much don't really know what to call it, how much lighted glass, there's some clinicians in here that will be able to correct me, but how much opacity is there and what those look like. So this is what we're trying to include in our model, but it's challenging, and we're hoping that we can get some of the Rona data because it's a bit more continuous in form. So here's just a quick exploratory look at the gene expression data. I just did a quick, what does it look like? Take the top 1,000 most variable genes, do a quick k-means clustering, and see how well that is actually clustering. And it's not really distinct clustering, but what I did find interesting is that you've got a lot of ISGs pulling the PCA this way. And we know from other work that we've done in the past that those sort of are associated with a more severe disease, especially in the mouse model work that I've done in the past. But when looking at the metadata and how this sort of goes back into the clusters, you can sort of see that things like cardiovascular disease, even the site, time point, and other comorbidities might be sort of associated with different clusters, which is important to keep in mind. This is why I always do stuff like this, so I can sort of get an idea. But when we look at if there's any sort of correlation between our clinical factors and our principal components, there's nothing particularly jumping out as super interesting. So, as I mentioned, we've currently been using high flow oxygen as a proxy measurement, which we know is limited, but it is a clinical intervention that is used, I guess, when patients are in a particular state. Yeah, and our other clinical outcomes were not suitable to choose because of limited power. But we've gone with it anyway, and we've tried to done some logistic regression to see if we can use this data to predict whether the patient needed to go onto high flow oxygen. And we've generated some quite consistent models, but this is very much still ongoing. And our next plan really is, instead of giving a classifier to sort of like look at, so whether that's ICU or death or high flow oxygen, we're going to go back to basics a little bit more and stratify based on their molecular phenotypes and take that more unsupervised approach in hope that we find either something that's targetable or that will help clinical decisions. So that was a very brief overview. If you've got any questions, come find me afterwards or ask me now, of course. And I just want to say thank you to everyone in the Dragon Consortium, CHOP-MD, Julian and Tess and Catherine for helping me do those samples, and the UK ICN for supporting me to get here. Thank you.
Thank you. Thank you for that talk. It's a very nice description of how to use analytics to identify biomarkers. So I think we have time for a few questions.
Thank you very much. Great talk. How do you control for your sampling times when you compare COVID and flu?
So I was the person that's just got handed the blood samples to sequence, but it's a really good question. And I think this is a problem in a lot of clinical studies, right? In this instance, there was, before COVID happened, there was already a flu point of care trial going on in the University of Southampton, I believe. I'm looking at Jim to correct me if I'm wrong. And the same group, the same working team, when COVID sort of emerged in the UK, took that opportunity to sort of match back to that cohort as a point of care trial. So it's the same sort of location. It was a different year, so it was the previous flu year before COVID came out, but it was sort of to try and compare that when a patient comes to hospital, what does their transcriptomic profile look like? And yeah, they were all like extracted and sequenced at the same time. That makes sense. Mm-hmm. Well, again, this is the complexity of clinical studies, right? Like, a patient might turn up to hospital on day one of having their infection, and a patient might be a bit of a slow burner and have their inflammation at like day 6. So it's really difficult to draw that comparison. So I always like to think of it of coming back to the clinical implication of patient turns up to hospital, and that's when I don't like, you might want to understand where they are in terms of like inflammation's a good one. Are they in a super inflammatory state, or is something else going on? Can we sort of look at their trajectory from there? But yeah, it's a huge limitation. And like with any clinical stuff, you can't know what everyone's been up to, and you don't know. So yeah.